so that we can continue knowing how everyone is feeling today. Okay, this is recording now.
Hello, it's Mark. Thanks for asking. It was a really full week, and I'm still trying to understand most of the basic concepts from our program.
Okay, and who is this again?
I'm Irmak.
Okay,
IRMAK.
Excellent. Well, thanks for letting me know. And I think today we'll kind of cover some of the same basic things. Hopefully, if you do come across anything that's unclear, you could pause me and I will be happy to expand if anything is not clear yet.
Okay, thanks. By the way, how are you doing? Like you, Ipsha, and all of the team?
I think we're doing great, except for Beeps that told me that she's not feeling that great today. Okay, so she's okay. So it looks like we're all okay. I'm doing great. I'm excited to continue this program and see how we can make the most out of the exercises that we will have online. And some of the concepts I hope will be much more clear after today's session. Again, if anyone has any questions or wants to have more explanation of anything that we go through today, this is the time to pause and discuss any of these things. So please feel free to interject and ask any questions. I'll also pause on specific slides that I have already prepared. If you have any questions there, you're welcome to post them in chat as we go along, or interject and we will stop. So again, what are we going to be doing in this session? We want to, last time we spoke a little bit about kind of general approach to processing data before it could be analyzed. And so today we'll move a little bit forward with that concept, and we'll talk about transcriptomic data specifically, so that we can explore how data visualization and different types of filtering and normalization steps really can help us understand what's in our data set. But before we do that, I wanted to mention that it's good to see the progress, and I think the chart also looks very nice. We can see clearly here that after our first meeting, some of you have probably completed the introduction to bioinformatics, and then we started talking about pipelines, and then we started talking about coding. And so hopefully now you're seeing how all of these things fit together. After today's session, I think you'll come away with a little bit more of an appreciation for the differences between these different aspects, because they truly are different. The educational part allows you to learn the logic and really understand why we're doing what we're doing and what are the expected outcomes. When we process big data, we obviously can try to do this on our computer, but there are just so many limitations to being able to do this effectively on a regular computer or even on a regular server. So it's really about having an environment that is set up to process, analyze, and visualize big data. But once we have the data set that we're interested in and we want to do downstream analysis, you can do the standard tasks by launching these preset pipelines or code blocks. But oftentimes you want to modify a little bit what exactly you're getting. You want to make the visualization more informative, or you want to do analysis in some way that was not thought of by the developer of a particular tool. So that's where coding really comes very handy. The objective of all of this is, of course, not to get stuck in the technical details, but to learn the logic, be able to execute preset analysis pipelines and methods. And then once you know what you want to do and how to do it, to be able to execute that on your own using Python or R, or even using tools like Excel, because there are powerful data manipulation software tools out there: Tableau, Excel, OpenOffice, Google Docs, or Google Sheets. So there's many tools, and no one said that one tool is better than the other. It's more of the use case. Why are we doing what we're doing that really makes the biggest difference? So this first part that we are in really is intended to get everybody started. So hopefully by the end of today's session, you will be comfortable with loading data and understanding where the data came from and what's in that data, so that you can practice in the session of the hands-on to work with different types of data sets. In part two, we'll talk about more advanced methods of analysis, because we can quickly run through a few examples, but to really understand the details of why we're choosing this method or that method, or working with this data set in this way or the other way, and what can we really get out of these different methods, requires some time. So that's when we will talk about some of these more advanced methods. But let's make sure that we first cover the basics and everybody is on the same page with what we are expecting to get from these different analyses. In our last time, we started talking about different types of omics data and what do they represent from a biological standpoint, as well as what type of data they are from a technical standpoint, because they are very different data types. So when you look at any new data type that comes in, whether it's an omics type of data or a non-omics type of data, you always have either an intuition. For example, if we're looking at business data or maybe some device like a smartwatch, that is very intuitive, because we can imagine somebody wearing a smartwatch and this is what heart rate means, or this is what their level of activity means. But when we look at molecular data, it's a little bit less intuitive. So we really need to understand why the specific wet lab techniques were designed and how that wet lab technique is translated into digital data. So that's the big challenge here that we're trying to cover. So let's do a brief review to see where all of this data really comes from. So the technologies that we are discussing, one example of such technologies is called high throughput technology, and it could really mean a lot of different things. For example, you can do scans or images of cells, and that would be phenomic data or phenotype data. You're really characterizing the shape and the structure of a cell externally. Once you go beneath the surface and use something like next generation sequencing, you are sequencing molecules within the cell, and those could be RNA or DNA molecules, or that's what next generation sequencing will provide us with. And it allows us to study that data at a few different levels. For example, we can look at variants, and I think last time we looked at how to create a pipeline of variant calling, so identify specific changes on the DNA that can characterize a sample. The other data that we can look at is epigenetic data, and epigenetic data could really be of a variety of types. For example, we can look at chromatin immunoprecipitation, or ChIP-seq. That data allows us to understand how tightly packaged or unpackaged chromatin is, or what types of interactions are there between different tags on the chromatin itself, on the histones. We can also look at whole genome bisulfite sequencing, which is an approach to sequence DNA and measure what areas on the DNA are methylated. We can also look at gene expression, isoform expression, expression of long non-coding RNA or microRNA, so different types of RNA that play a regulatory role. And that regulatory role can allow us to understand gene expression levels or reasons for variation in gene expression, even though oftentimes many of these non-coding RNAs are not really known for their function exactly, but we kind of have a sense of the mechanism, so we can try to piece that information together. In any case, the objective of that process is to prepare structured data. So prepare a table, and that table allows us to then do all kinds of data analysis. So we can then work with the structured data, and we'll talk about a few different things that we can do with this data based on what type of data this is: categorical, numerical, continuous, etc. So these are some basic types of data that we can have. And one of the things that we talked about last time is that bioinformatics, what's unique about it is the combination of our understanding of the biology as well as the data part of it. For example, when we measure gene expression, we need to understand what genes are, we need to understand how they are organized, and we also need to understand what types of changes in the sequences or the level of expression really play the role that we are seeking to interpret. They might cause a downstream effect, direct or indirect effect on the phenotype. And so as we talked about different types of mutations, we also talked about other resources that you can leverage to understand these resources in greater detail. This program is not going to be focused on any particular type of an omics data, so we're not going to discuss extensively what mutations mean or how to measure them or what are the analysis methods, but if you have the time and the drive, you can definitely explore Introduction to Genomics, Genomics 1 and Genomics 2, that talk a lot about how this data is prepared and analyzed. We also talked about mRNA data, or gene expression and alternative splicing. And there we talked about the process of transcription that is a proxy for measuring translation and presence or absence of different proteins as well as their abundance. It's an indirect measurement, so we need to understand that we cannot directly infer from here the outcome of this process, but it is a process that has been used as a reliable way to estimate mRNA expression. And so it's also a useful data type that we can study using different types of data analysis methods, including machine learning. And we'll talk today about one use case in breast cancer that allows us to connect the data at the molecular scale with clinical scale to really understand what is the relationship between a clinical phenotype or a subtype of breast cancer and a molecular data set that includes gene expression levels. And we'll talk about some of the challenges as well as some of the opportunities in that example. So we also ran a pipeline, and a pipeline, I'll talk a little bit more about this in a second. A pipeline, really all it is a series of steps that take in some input, then convert it to an analyzed output and pass it on to the next step. So as you remember, we looked at how you can load in raw data, and we'll talk a little bit more about what is that raw data today. We then process the individual pieces of raw data, no matter where they came from. We just take a look at each individual piece and make sure that it's accurate and does not contain any technical additional information that is not useful for our analysis. Then we want to organize the data. So instead of taking a random bucket of reads, we try to piece together the puzzle, and we want to place each read in the best position on a reference. So we can kind of put together the pieces of the puzzle and understand which pieces do we have, which pieces do we have a lot of, and which pieces we don't have. So after that, we want to quantify how many and what kinds. And so for that, we have two different processes. One is the process of annotation, because all the positions on the genome are annotated, whether they belong to an exon, an intron, and how those different exons might be pieced together to form a single isoform or a transcript. And we also want to quantify. And we talked a little bit about how it's not a trivial task to quantify. So first of all, we want to quantify a gene which consists of multiple exons. The data comes from multiple cells. We're not taking single cell data here, we're taking bulk RNA-seq data. And ultimately, we also want to incorporate some knowledge of quality and abundance of source material, like the source RNA that was taken, and adjust for all of that variation. So it's really something that requires some computational capacity. And then as we go through the details today, you'll also see a number of resources that are available to go deeper into RNA-seq data, and that includes transcriptomics 1, 2, 3, and 4. For example, if you're interested in statistical analysis, which we will do in some of the other sessions, or you want to understand more about machine learning for transcriptomic data, you can use transcriptomics 2 and 3 to really independently go through all of the details of those analyses. As I mentioned, this is bulk RNA-seq data. And when we come to single-cell RNA-seq data, the challenge of how the signal varies between independent cells and how to connect all of this data together in a single comprehensive image has additional challenges, and those are explained in Transcriptomics 4. So for those of you that are interested to kind of explore this domain further, I recommend completing those courses, and then we'll talk about some of the additional projects that you can practice with after you've done that basic work. Now, finally, we talked about processing metagenomic data. And so even though the approach to generate the digital data here exactly is the same, we always sequence cDNA, even though we might take DNA or RNA or ribosomal RNA, every time the process is going to be analysis of sequences from cDNA, which is just a more stable form that you can use to generate sequences which are more accurate. Now, when we deal with metagenomic data, this data helps us not study a single organism, but really to understand the diversity of multiple, many thousands of organisms. And the challenge is that these organisms are different at different positions of their genome. So some positions are conserved or very similar. Some positions are many changes that are too many to count, right? So each individual might be very different from another individual, even though they belong to the same group of organisms. So one of the approaches to understand that kind of variety is to use operational taxonomic units, which are proxies for taxonomic classification markers that are based on different hypervariable regions in the 16S ribosomal RNA. So we'll not talk about this in too much detail. There's again a course on this, but the process of exploring that data also requires certain structure. And so the structure is done by analyzing, first of all, mapping instead of on individual genomes on a whole set of genomes, which are stored in a database. So we need to also access that database effectively. And then we can prepare a table as a result of this. And the table could be visualized and analyzed statistically. So here we also have a series of courses: Introduction to Metagenomics, Metagenomics 1, Metagenomics 2. And now we're also working on Metagenomics 3, which is going to include analysis using KIND for those of you that are familiar with this domain. But let's talk about what is the output data from each one of these analysis steps. The first one that we talked about is DNA sequences. So here's an example of that output from somatic analysis. The somatic mutations are mutations that occur as the cell divides. So it's not passed between parents and children. It is not inherited, but it is acquired. And that acquisition of mutations could happen because of age or because of some deficient mechanism to make sure that the DNA is replicated with fidelity. So there are a lot of processes that are involved in that. And one of the problems is that once that mutation acquires a certain level of cells that keep and maintain that mutation can cause significant changes in the phenotype. So typically when we look at tumors, one of the driving forces behind those tumors acquiring their characteristics, like cell adhesion, changes in cell adhesion, and how they're able to move throughout the body and metastasize, those are some of the dangerous processes that we want to detect early on. So the process of detecting somatic mutations is a standard component of tumor genomics or cancer genomics. When we look at the somatic mutation, it's a process of filtering out all of the mutations that we can find, or all of the differences that we can find in a given set of reads from the reference genome, by also accounting for the normal control, because we know that each individual is different from the reference in some ways, but the somatic portion is all the mutations minus the ones that are recorded in the healthy genome of that individual. So that's why we have this special somatic variant calling process, or a pipeline, that accounts for both of these sources of information. As a result, we have the actual somatic score for each position. So you can see that the positions could be of several different types. We discussed that last time. Those types include 3 different types of substitutions, because we have a total of 4 types of nucleotides. So 3 could be substitutions, another one could be an insertion, and another one could be a deletion. So a total of 5. And then based on the number of reads that we have and the type of mutation that we observe, we can calculate a score. And this score could be used, this is a score for an individual sample. So if we have scores like this for many samples, again, we have some sort of continuous data. That data could range anywhere from 0 to 225. So we can use all kinds of filtering, threshold, statistical analysis, and machine learning techniques to start differentiating between different types of mutations or different types of samples, and understand how the samples represent a condition or a phenotype, and then study what is the association between an individual mutation or multiple mutations that causes such phenotype change. Are there any questions about this type of data, the genomic data? Okay, if you have any questions, feel free to add that to the chat and we'll talk about this a little bit. Okay, so we have here key elements of this data set. We have the DNA position and we have the somatic score. So if you include here the sample name instead of the somatic score itself, you might have a matrix that could be analyzed further. Now, the other type of data that we looked at is RNA-seq or gene expression data. So gene expression table or table of genes right here and samples allows us to understand the relationship between level of expression and the type of sample. So we can understand if we have groups of samples, we can understand how do genes characterize the different conditions. We can also work with the genes, so we can understand which genes are more similar to each other or how they are different from each other. And we can also study regression, so relationship between different changes in phenotype and different changes in gene expression. So multiple types of analyses could be done with this table, but we need to remember how this data was prepared. And we need to understand how the numbers in these cells could be analyzed so that we incorporate our understanding of the technical and biological function that each one of these numbers represent, and how they could be different because of different factors. So the expression level is the key here, but the genes could be transcripts, they could be exons, so they could be a variety of things, and the samples could be individual samples or conditions, or they could be continuous or time course data. Now, the final data type that we talked about was OTU tables. So OTU tables represent some approximation, which is an average between multiple sequences. These OTUs range from 100 to 300 nucleotide long sequences. So these are actual sequences that we can later on find. But the sequences are related to each other in some way. And so they are related to each other, and they are related to a taxonomy or a group that we can place them in. And so that's why looking at this table by itself, we can use it as a reference in a similar way like we use the gene expression, only instead of genes, which are responsible for specific functions, these OTUs are belonging to different taxonomic groups. So in addition to having this kind of a table that shows us some abundance numbers, we also need to annotate OTUs by different levels of taxonomy. Okay, so taxonomy helps us select one of the levels of taxonomy, for example, by family, order, class, or phylum, and then look at, okay, if I have this phylum level of taxonomy, what can I say about diversity of a particular sample as compared to another sample? Or I can look at one of these taxonomic classifications like firmicutes and ask whether firmicutes are more or less abundant in a given sample from other samples. It makes it also challenging that we don't know what exactly was sampled, right? So there's a component of noise there just from sampling, and also a component of noise just from the way the data is prepared. So how it is technically prepared, whether it was sequenced in the exactly same way, and whether we have the process of assignment to that taxonomy in a similar way. So we typically approximate the absolute numbers with a proportion. Instead of looking at this is 5 versus 125, we would say 5 is 2% of the full number of units that we have, and 125 is 3%, right? And then we look at how significant that difference might be. So today, we'll talk a little bit more about gene expression data. In all other aspects, it is very similar to these other types of data. It's just that every single type of data that we talk about comes with this history of how the data was prepared. And when we talk about making reproducible and clear representations of our analyses, these steps have to be included. So whenever you say, I took this gene expression table, that's not enough. You have to say, I took this gene expression table that was prepared in such and such a way from these types of methods of data preparation. And that's why it's very important to understand the whole process from beginning to the end. You can't just say, I got a table and this is what I found in this table. Knowing that you understand how the table was prepared really gives validity and significance to the way that you do your analysis and present your analysis results. So when we think about the downstream steps, once we've established that this is the data, that's what the data represents, this is how it was prepared, we can start looking at patterns. Patterns means that we are looking at specific variation, and that variation could be interpreted in many different ways. So before we jump to interpretation, we typically want to understand the variation that we observe. So we really want to understand the data. A lot of times there are kind of these standard approaches to what we can do. And one of the standard approaches for gene expression is to do what's called differential gene expression. So differential gene expression means that we group our samples by condition and we compare the differences by condition. And we use all kinds of techniques to eliminate variation within condition to understand what are some significant genes. Now, this type of a condition is this type of an analysis, is a very biologically useful way to speak about data. Because from a biological perspective, you really don't care as much about an individual sample as what that sample represents. And the whole idea of biological experimentation is focused on replicates and repeated experiments to eliminate that kind of technical variation between individual samples. But in the modern time, in the time of this kind of sequencing data being applied to real world data that was not prepared as a result of an experiment, we face a completely different challenge. So that's why this paper is a useful example to understand what role bioinformatics plays in biomedical discovery. So when you think about data science, one of the reasons why data science has become such a prominent skill expected from bioinformaticians, biologists, clinicians, technicians, etc., is because we are living in the world of data that was prepared differently. What does that mean? When we think about an experiment, you can control a lot of the variation that you cannot control in the real world. So outside of the lab, you cannot say, I'm going to pick 3 people that are exactly the same, have the exact same cancer, and that cancer is at the exact same stage. That's just practically impossible. So what you have to do is you have to deal with that variability. In this case, we have a publication that took cell line data. These cell lines were derived from patients, grown in different types of conditions, but sourced from different experiments. So this simply represents a derivative from a real patient that we can start analyzing. The objective of the paper was to evaluate 2 different things. The first one is what type of data is most informative to differentiate between different patients. And number 2, whether a particular type of data or combination of some types of data could be trained, could be used to train a machine learning model to predict how they would respond to treatment. So I want to make sure that this is all clear to everyone before I continue. Let me ask you a few questions. What is meant, so let's just take a look at this paper together, and I want to ask you a few questions just to understand that everyone is on the same page. So this is the link to the paper. And I'm going to open this paper also. Sorry. Okay, so let me know once you've opened this paper. All right, excellent. So let's take a look at the at these first two paragraphs. First generation molecular profiles for human breast cancers have enabled the identification of features that can predict therapeutic response. However, little is known about how the various data types can be best combined to yield optimal predictors. So let's decompose this sentence. First of all, let's think about what is first generation molecular profiles. So any guesses about what this means? Okay, what is molecular profile? So what are we talking about here in terms of profile? Okay, Sanger sequence maybe generated by NGS. Any idea? Any other guesses or ideas? Microarray not generated. Okay, excellent. So when we talk about molecular profiling, we can talk about a number of different molecular signatures. For example, immunohistochemistry. Okay, immunohistochemistry is the ability to stain cells based on expression of proteins. And this is a standard clinical procedure. So if you go and have biopsy, that biopsy is a piece of tissue. The tissue could be put into a machine that is tested for using different antibodies that have color attached to them. It can demonstrate that these cells have a certain protein on their surface. So immunohistochemistry, or short IHC, is a technology used to determine the level of protein expression in a tumor. So this is that first generation molecular profiling. It really allows us to focus on certain types of markers. It is very selective. Then you have additional types or variations of the same, for example, FISH or fluorescence hybridization. It also looks not only protein, but it detects gene deletions, amplifications, translocations, and fusions. So it gives us better characteristic of what's happening. We can look at next generation sequencing. Next generation is second generation, not first generation, but it's the next generation after that. And so that is an ability to really study DNA and RNA variation. We have Sanger sequencing, which examines strands of DNA. Typically, Sanger sequencing would be applied to targeted sequencing since we know where it's coming from. There's other types of analyses as well. Pyrosequencing, quantitative polymerase chain reaction, or qPCR, fragment analysis, which also detects changes in DNA or RNA. So there's really a number of different types of treatment. Next generation sequencing is not the first generation sequencing, but both of these approaches for molecular profiling, which is the important part here, is to be able to detect changes at the molecular level. And that change could be variation of gene expression, but it also could be mutations, methylation changes, protein changes. All of these are molecular signatures. So what are the features? Let's just list out what are the features that we're talking about. For example, Glory also mentioned level of gene expression. So gene is a feature. What other features can you think of when we discuss these molecular profiles? So gene is a feature. From level of gene expression, that's the data. Gene is the feature. So what other features can you think of? Okay, protein, mutation. So mutation is a characterization. This is a mutation, this is not a mutation, right? But what is the feature? The feature is the position on the genome, right? So that nucleotide, that single nucleotide could be a mutation, could be a variant, could be a SNP, could be a regular, what we see in the reference, right? So the position itself, protein itself, right? So those are the molecular features. Let's also think about prediction of therapeutic response, or what is an optimal predictor. So what do you think is a predictor in this case? How would you define a predictor? Any ideas? What might it mean that we say predictor? Okay, response. So here predictor is used. Well, drug response is what we are trying to predict. That's the outcome. But the predictor itself means that we have identified a molecular feature that has found to have a strong association, statistical significance in terms of association with the outcome, right? So it's measurement of the association. So when we say predictor in statistics, we are saying that that association is statistically significant. When we say a predictor in machine learning terms, we are meaning that that feature contributes to the prediction of the model that we have trained. Does that make sense? Okay, so when we speak about this process of comparing different types of features, molecular features, to understand their relationship with the outcome, we are defining the problem statement. So let's take a look a little bit at this whole field of biomarker discovery. So as people have started dealing with this kind of data that comes from patients, different techniques allow us to understand how the specific genes or mutations or methylation patterns are associated with outcomes. So a typical approach to that is to look at level of recurrence or the survival, right? So this is a survival curve. So you can see here RS, that's rate of survival, or ROR, which is the recurrence. So rate of recurrence. So here you can see different groups forming over time. And we see 100%, so this is the 100% of patients. This is the years that you have for these patients. So if I take a point anywhere on this curve, for example, here, this is after 4 years, and about 7 or 85% of the patients have died, right? So the survival has went down. So as I look at this curve, and I understand that based on, so this is based on PAM50, which is a 50 gene panel that measures the level of expression of a gene, I can predict if I measure at a time point where I have significant separation between these three groups, I can predict what will happen in 10 years, right? So if I take that here, I don't have to wait. I know which group a different patient will belong to. So this is the prediction. That ability to predict is an essential part of biomarker function. So when we speak about these features that are associated with some outcome, we are talking about a molecular indicator that could be used as a biomarker. So one of the challenges that data science solves in biomedical research is identification of such useful biomarkers. Now, out of thousands of genes, we have here two examples of panels, and panels mean sets of genes. So PAM50 is 50 genes, Oncotype DX is 35 genes. So we have different combinations of genes or different combinations of features that help us inform the prediction. Now, here you can see that the difference is in how close to each other these curves are. The closer they are, the more likely we are to make a mistake. In other words, if we see the measurement at, for example, 3 years and they are well separated, we have high confidence in the prediction. If they are still close together, like here, maybe at 2 years, it's not a good predictor to take that sample here and to predict what happens in 2 years, but it is a good predictor of what happens in 3 or 4 years after that. So this is one use for such feature analysis. The other useful information that we can extract from this data is how to go about decision making. So when we make a decision, a clinical decision is the interaction between a clinician and the patient, and we can take a look at different levels of risk that would inform that decision. For example, if you are saying, I really need the money, and you're deciding, am I going to find a job or am I going to rob a bank? So how much do you want the money, and the risk associated with robbing the bank might inform your decision. Maybe that's a bad example, but it's an example that came to mind. Now, if here we have treatment decision, we are also looking at the level of risk, because each one of these therapies has an associated risk. Chemotherapy, for example, is known to produce mutations and all kinds of side effects, lowering the quality of life and leading to potentially very bad downstream kind of condition changes that might need all kinds of additional treatment in the future. Radiotherapy, the same. It also depends on how effective it's going to be. And surgical therapy also, a good example is when we have metastasis already happening, maybe surgical therapy is not going to be as effective, right? Because the tumor is already metastasizing. So this is one component of treatment selection. So where we put these patients on this curve and how much risk is carried with each one of these treatments helps inform that decision. The other part of it is that the drugs themselves are changing. So first of all, we can kind of generalize and characterize our patients or stratify the patients, putting them into different groups. We can also take a look at stratification of risk, and we can also look at the specific molecular information that we have about the pathway or the function of these drugs. Now, some drugs, for example, some standard chemotherapy is just generally reducing the replication rate. So it's going to reduce that rate of replication in any cell that it affects. Other drugs are targeting a very specific pathway. So a good example of that is a drug called Herceptin. Herceptin is basically an antibody that blocks a receptor called HER2 receptor. So it's a growth factor. This growth factor connects the outside of the cell, so it could be easily accessed from the outside, to a pathway, a signaling pathway that tells the nucleus to produce certain type of mRNA and proteins, and so it activates certain type of molecular activity. If we block that receptor, we're going to stop that activity. And so now when we are in this area of targeted therapy, we are talking about drugs like Herceptin. Another type of a drug like that is Opdivo. And those drugs really have a very specific molecular target. Now, the difference between tumor cells and non-tumor cells help us decide whether that's going to be an effective way to intervene specifically for tumor cells. So the delivery aspect here is also very important. Now, where does machine learning or data science fit into all of this? we are originally starting from something that looks like this, right? Or something that looks like this table. So we have a gene expression table, and each gene has a specific pattern or a signal that it carries. And we would like to find those types of genes that help us identify different types of groups based on clinical information that we have about these patients. So let me ask you if maybe you can let me know if that was clear, if you understand the concept before we move on to speak about what we technically do to start this journey of analysis of this type of data. So let me know if you have any questions or if anything from what I said was not clear so that we can clarify that now. Okay. So if there are no questions, let's move on to the technical part of the story. So first of all, let's talk about measuring mRNA abundance, right? To detect something, we need to be accurately defining what the signal is. So when we're measuring mRNA abundance, we're looking at a way to count how much of that RNA is present in a sample. And like we said, there are many different ways to extract that level of information, and those are different types of techniques. So the first generation techniques would include quantitative PCR, qPCR, microarray, FISH, and others. And then we come to this next generation sequencing idea where we can just shotgun sequence all of the little pieces of RNA, and we have to glue them back together. And so we have to use these pipelines to process this data. As a result, we have several different files that go into a single pipeline. FASTQ files that contain the reads and the quality score for each position, FASTA, which is the reference genome, it's stored in a FASTA format, and the GTF, which has the actual beginning and end of each one of our features, genes, exons, transcripts, etc. So then the process of gluing it all together is directly correlated with the process of preparing the data. How is the data prepared? First, those reads are prepared by sharing the DNA. Adapters are attached, or ligation of adapters. So those are ligated, those are attached to these short pieces of cDNA. Then we have the flow cell, which is basically a little plate, glass plate that sits with PCR amplification that could produce enough material for sequencing. And the sequencer produces signal based on different colors when different types of nucleotide adapt or connect to the sequence itself, and we measure the different colors, and then we can say whether this is T, C, G, or A. So as a result, you have a sequence, and that sequence contains all of the information that was prepared right here. So that is why we go through the process of, first of all, pre-processing, then mapping, and then quantification. So if we look at the logic behind this analysis, we want to make sure that the data is clean. We then want to identify where it belongs on the genome. Then we want to quantify. Because we're speaking about a data science program here and not standard bioinformatics, maybe knowing just the logic is not enough. And maybe some of you are wondering, well, how do I do this in maybe R or Python? So technically speaking, if we look at this process, you're speaking about calling a particular function or an algorithm or a script, which is a program, using parameters that are defined within that program. For example, a typical way to call so HT-seq is a Python framework to work with high-throughput sequencing data. Here you have other types of analysis methods like RSAM, SAM, and Callisto, and all of them typically would be called in what we call bash script, so a script in Linux that will allow us to install and then call a particular function from a library of different functions that are included in a package. So that is the process that we're skipping over here, because we want to get to the data science part of things. And so the data science part of things starts when we have this table of expression. And that we are starting to view this not as biological information anymore, but we are looking at this as a source of data. So the first thing that we want to do is we want to understand what is in our data. To do that, we can visualize data. So that's one example of what we can do. And this example shows us that we have different levels, and we can start saying, well, is this level higher than this level, or is this group higher than this group? The other part of it is we can start doing transformations to this data so that we understand what is inside the data as a whole instead of individual ones. So if we have thousands and thousands of genes, how do we actually find which genes are informative, and how do we actually understand overall what do we have in our data? So here we have an analysis, kind of a visual analysis. So these are box plots. What do box plots show? They show the maximum of the 75% of all data, the minimum of the 75% of all the data, and what's called the interquartile range, as well as outliers. So this is a visual summary of all of the thousands of genes that are there in our sample. As you can see, before the data is transformed, normal data, so without any transformation, it is difficult to see. It's difficult to understand what's inside the data because the outliers are so strong. So these outliers distort our data. And this distortion, even though here we're looking at it just visually, it also affects the way that we can infer, statistically speaking, the differences. Why? Because if I have a gene that is 16,000, and I have a gene that is 2, 3, 4, or 5, the difference between 16,000 and 2, 3, 4, or 5 is so strong that I wouldn't be able to understand how significant it is in the sample overall. And so to address this problem, we can do all kinds of tricks, really, with the scale of the data and the way that the data is organized within each sample, trying not to really change the values themselves, but trying to manipulate the way we look at the data, going from this angle, from that angle, from down at the bottom, from at the top, to really get a better view. So today, we'll take a look at how to do this first in R and then in Python so that everyone can practice a little bit. But just to make sure that everyone understands what we did before this, let me ask you a couple of questions. What type of files are used as input for the RNA-seq pipeline? So number one, FASTA or FASTQ files, which include DNA sequence data. 2, SVL or URL files, which include RNA sequence data. 3, RSAM or TXT files, which include DNA sequence data. 4, Excel files, which include RNA sequence data. So which one do you think is the answer for this question? So you can put the result in the chat, 1, 2, 3, or 4. Okay, so I see that there's a problem. The correct answer is number 1. Okay, Gloria actually got it correctly. So the actual files that we put in are the FASTA or FASTQ files, because to analyze the data, we need the sequences. What are SVL or URL files? They're not files, they're just links. So they just help identify where these files are stored. And why are we not using RSAM or TXT files? We're preparing those files, right? So the answer here is FASTQ or FASTA. Does that make sense to those of you that answered 2 or 4? All right. So the reason why we're using cDNA is because the RNA is a less stable form, but we can easily convert it to the DNA form. And when we convert it to DNA form, it is more stable, so the sequencing is going to be more reliable. So the second question, which of the following are pre-processing algorithms? 1, top hat and DSeq, 2, PCR clean and trimmatic, 3, both type 2, BWA, or 4, edge R and DSeq 2? All right, excellent. So most of you are correct here. It's PCR clean and trimmatic. So the pre-processing steps, what do these methods actually do? The PCR clean removes PCR duplicates because PCR amplification, it can be biased just based on the sequence. And trimmatic cleans the technical sequences that are adapted or attached to those read sequences, which are the actual data. So now let's talk about how can we analyze the downstream, how can we do some of these downstream analysis steps? And so we'll look at the following examples. So please go with me to code.omicslogic.com and we'll do the following steps. We'll make a histogram for each one of our samples. We'll learn how to filter for low values, change the data to logarithmic scale, and make a bar plot and a box plot, just like we saw. And we'll also introduce how to do Python for some of these steps. Harpreet? Harpreet, are you there?
Yes. Hello, Ilya. Yes.
Hi. Okay, so are you ready?
Yes.
Okay, so I'll pass it on to you.
Okay. So, hello everyone. So, are you able to log into this Code OmicsLogic platform? So, just let me know so that we can do it together. In the last session, we learned about how we can upload a file in a R. Now, let's go one step ahead and let's see how we can manipulate our data and how we can visualize our data.
So before we go on, if you are already on this lesson, data visualization, let us know. So let's just wait a couple of minutes so that everybody is in there. If you're logged in, maybe go back to all the courses, Harpreet, so we can see how to find this course.
Yes. Okay, just let's go over there. Let's start with, once you are logged in, so then here you will find the courses. So if you click on the courses, so here let's do it first. Let's do it in R. So just go to this course, that is Introduction to Data Science. So in this tutorial, we are having multiple lessons. So let's go to the data visualization. So if every one of you or any one of you able to reach to this interface, just let me know and just post your answer in the chat. Okay, that's great. Okay. So before doing analysis, let me share a data. So I'm sharing the link of a data. Just take this link, and so that we can proceed. So what is the special about this data?
One second, Harpreet, it looks like Anita doesn't have access. So Anita, if you could, these are actually free courses. You should have access in any case, but if you haven't registered on the site, you can just register. And for now, even if you have the free account, you'll have access to these courses.
Okay, so meanwhile, I just let you know about this data. So it's like breast cancer cell line data, where we have gene expression in the row and our samples are in the column. So apart from that, this particular sample data is actually containing NA values. So like here, we can look at here, if I'm looking at this data. So if you look at this, there is NA value. So similarly in this data, there are some of NA values. So now today we will understand first, of course, we will start with how we can upload our data in R. Then we will how we can remove these NA values, and then we will also do how we can do the log transformation in R. So and then how we can visualize our data in form of box plot, in form of histogram, in form of bar plot. So now let's. So Anita, are you able to log into this server? Okay, so as every single lesson will explain, always started with the first step, that it will provide you the introduction. So like why we are doing this tutorial, and what are the different steps that are we are going through during this tutorial. So it will explain you every single step. So if you what kind of data that we are taking here, so all those things. So again, like why this is data cleaning step. So what is this data cleaning step? It shows you first how we can actually remove a column or remove a row from our data frame. It will explain you. Then next is the data cleaning step. So it is like how we can remove NA values from our data. So then apart from that, how you can apply the log transformation. So these kind of explanation is given in all these steps. So once you complete it and you understand regarding all these steps, then we can go to write our code. So now let's start. First of all, just upload our file. So last time I have told you that how we can read a file in R, that is with the read.table command. So then I can provide my file and path of my file. So this is code breakdown. So it will explain you every single step. So once we complete it, now let's go to the playground where we can write our own code. So now let's start with what we are doing. I'm just commenting every single thing here. So first of all, let's upload our data. So how we can do it? We just need to read.table. Then in the double inverted commas, we need to provide the path of our file on which we are going to work. So as I have already copied that path, and then I have to define whether my file has header or not. So how I can do it? Header equal to true. So then I have to define how my samples are separated or the columns are separated. So I can define it by separated as this in this particular file. My columns are separated by the tab. So once this file get uploaded, now how we can check this file, whether this file is either you can print whole file. So here other thing we can do, we can even check how we can visualize, how we can check that how many columns and how many rows are there. So this we can do by just write one thing, that is dimension dim. With the dim, if I am just defining, and I can even check how many columns are there, and I can also check whether my file get uploaded or not. So just let's do it. Just with the dimension, I will understand whether my file is uploaded or not. So I think I need to just upload this file again, refresh this page again. Might be some. So just in case we sometimes we need to refresh page. So now let me just do it here. So I just need to define the path of my file. Tab separated, then I have to define header, that is true. Just close it. So let me just do it. So then I can, okay. So head, with head we can visualize is top our top lines that were in file. So apart from that, I have told you that with the dim, that is dimension, it will let us know how many columns and how many rows are there in the column. So if you do this, so it will initially tell you that here we are getting dimensions. So it is like here are the at the first it will give us the number of the rows. So it indicating that there are 1000 rows and there are 39 columns. So once you do this, so then in the next step, so then we will remove our so next step that we are going to do, that we are removing our first column that is containing IDs. So we are removing it here with this command. So if you look at now, look at this dimension there. So how we can do it? So okay, so like here we can check whether the column is get removed or not. So it is like first in the square bracket you have to define the comma, then you have to define the column that you want to remove. So like here -1, so I'm just removing my first column here. So once you able to do this, so now as we know our data contain NA values. So how we can do that? So let's remove the first of all, you cannot plot your, I mean, even if I try here, like here, I just want to do the bar plot. I want to visualize my data in form of a bar plot. So it will give me error as there are the NA values. So if, sorry, before that, I think we have to define it as a matrix, df as dot matrix, to plot any graph or any plot like bar plot or box plot in our first we as they are these are the data frames. So first of all we need to convert them as a matrix. So how we can do that? That is, if you write like as dot matrix, then you define the data frame to for which you want to convert. So if I am just doing it, so this doesn't show any result. The reason is like that this particular data has NA values. So the first challenge, we need to remove those NA values. So how we can do it? So if we look at this lesson, so here we understand that with the exclude dot NA dot exclude, we can remove our NA value. So now let's do it here. So let me just make a new data, that is I'm just defining data 1. So now with NA dot exclude, we can remove the NA values from this data. And we can also check whether these row can detect it removed or not, again with the dimension, that is with dim. So we can check now the dimensions as well. So now let's do it. Okay, so here what you get, so finally initially you have 1000 rows. So here how many rows we able to get it? With this we have removed all those rows which contain NA values. So we are left with 988 rows now and 38 columns. So now the next step, how we can actually log transform this data. So let me do it like, this is very simple. Once you got data, no, just with one command, that is you just, no, I have to define, this is log normalized log transform data. So I'm just naming it like log underscore data. So then I can do it by log df, log data1, as this data1 is the data from where I have already removed my NA value. So let's do it. Okay, so how will I know that whether my values are transformed or not? So one way is to do that summary. So let's do the summary for our initial data. So it will, summary will give us like minimum value, maximum value, right? And apart from it also give us first quartile, third quartile, all those kind of information. With the summary command, we can get that information. So like here, I'm just, first I'm doing with the data one, which from, which is not log normalized. So again, then I am finding those summary statistics with the, from this log normalized data. So log underscore data. Okay. So now, if I am just commenting this a first part. Now let's see what are the minimum and maximum values for this. So here we are getting like, if you look at this. So here we got it, our minimum value in our data is 0 and maximum value is 11.86. And this is what the maximum and the minimum values. Then we have first quartile value, then we have the mean value, that is 5.27 and 5.11. So now let's find for our log normalized data these values. Just I'm commenting. So if you look at here now, minimum value again, infinite value, as if you take the log normalization, why it came with the minimum value is 0. So that's why it's giving us infinite value. So as even in the Excel, when we are log transforming those values, we need to add +1. So when I am doing the log of this particular data, so I need to add 1 +1. So now if we look at this, okay, so now you get the minimum value is 0 for log normalized data and maximum value is again 2.54. So this is how you can simply, with just a one step, how you can log transform your data. Here we just, what we did, we have just did the log, and then in the data one, we just added +1 value. So this is how you can do this only in R. So next step, how we can visualize this data. So let's do it with the box plot. Now let's first draw the box plot, as we have already already told you in this session that with the box plot we will get the summary statistics about the data. So that with this summary, as I'm this I'm just getting for this particular data using summary. So now I can visualize using a box plot. So if I'm doing log underscore data, so now we will able to plot a box plot. So if you look at this data, so here you're able to get the log statistics or log box plot of this data, and you will what are these values? So this, at this value, specifically the dark in this box plot, if you look at this dark black color, it indicated your median value, it indicating your first quartile, it indicating your indicating upper quartile, it indicating your lower quartile. Then you have the outliers. So this is like minimum value and this indicating maximum. So as apart from that, it also giving us the outliers. So this is how you can visualize your data in the R with very simple steps. So are you able, any one of you able to produce the box plot? So any one of you able to just draw a box plot for this data? Just write yes or no. Okay, that's great, Amika. Okay, that's great. So are you people also able to do log transformation? Okay, that's great, I think. So the next thing that we can do it, once this is a box plot, now we can also draw the bar plot. So this is like we are just doing with this data only, log underscore data. So this is how we able to obtain the able to produce this the bar plot. We will also add the colors to it. So I'm just at starting, I'm just doing a one step that how we can.
One question, Harpreet, one question. So which specific gene is this?
Okay, so I have.
So the difference between the two, right? This is a box plot of all of the genes for each sample. And maybe you can do CX equals 2 to make sure that your X columns are rotated. And then for the bar plot, you want to select just one of the genes. So maybe you can show, you know, like gene 1, gene 2, gene 3.
Okay, I think I can select one column. Oh, sorry, one row. Not able to put it. The first row, if I got it.
Maybe now change it to 3, 4, etc.
Okay. 3, 4. For the first row, this is when the fourth row that I'm taking here. So apart from that, I think it's a similar. I'm something missing here, I guess. Let me just correct it. Just do it. Just need to. The color is already defined. Histogram. So I think,
Yeah, you forgot the T there for histogram.
okay, I see. Okay, I just. But just again, we have to just define log dot data, data. Color is equal to red. So this is how we can obtain a histogram, but it is giving, I think, histogram of this particular sample. So with the color, we can define the color. I think here, Ilya, I'm missing something here for particular bar plot for the selected sample. If I'm doing this bar plot for the second row. So like here, I want to. So this is how you can do it. Does the removal of the first column have some relation missing the gene name? Yes, I think, yes, we can say that. Even we can do it like, if we don't remove the first column. So let's take it like df2. So let's just do it like this only, df2.
Oh, okay. So what you're trying to show is a few different things, right? So you're trying to show here how we can remove the NA values. That's one thing that you're trying to show.
Okay.
And the second
Yes.
thing you're trying to show is how to prepare the data just so that it's only numeric data. So since you have different values, you want to just create numeric values. So those numeric values can then be visualized. So if we go through the exercise, we can see how, first of all, we can load the data and get rid of anything that is not numeric. That's number one. Number two, we can then look at how we can visualize the numeric portion of the data. So if we go through this code, and maybe I can just briefly share my screen here.
Okay.
Okay. So here we have, okay, for example, we do this is an example where we take our data set. We load the data set. This is just a smaller data set than one Harpreet is using. Once it is loaded, we can then take each one of our genes and we can visualize them. So if I remove the logarithmic scale, it will look, and I can, by the way, save this just so I have it for reference. And let's compare to what I had before. Okay, so we can see that the scale changed. So here I have from 0 to 8, here I have from 0 to 2. Right, so the logarithmic scale reduces the scale, and you can see that the variance, so the lowest, are going to be closer to the top. So that's what logarithmic scale does. This is for individual genes. So what if I do it as a box plot? So I can just do box plot of df and box plot of df log. So let's see what happens, and I can comment this. So here's my box plot, and here's my box plot. Now this 15 gene data set that I'm using right here is already normalized. So that's why the difference is actually not that great. When I see an overall scale from 0 to 12, that already tells me the data is normalized in some way, because typical gene expression data, as we saw, goes all the way to hundreds of thousands. So here in this example, if you actually use this data, let's just run it again.
This is also I think log normalized data more or less.
Yeah. So instead, you should take an example from the actual, big data set. But here, the difference is more, right? So the difference here is more visible. But in the future, Harpreet, when you do this presentation, you show a non-normalized to normalized. That would be the effect that we want to produce here.
Okay.
Okay. So the idea here is to be able to show visualization. Let's quickly, I know that it's time to complete this, but I'll just quickly show the portion with Python so that people can get started. So could you all please go to data visualization in Python, and we'll make sure that we can all get started here doing Python as well. Here you have some examples of how data could be visualized, filtered, and produced as a visual. So let's go to complete and go to next. And Python works a little bit different in this OmicsLogic playground. So the way you do it is you have to download this to your computer. So you download the linked file. Once that file is downloaded, you just drag and drop it right here. So we'll actually load it into the browser. So you can't use the HTTPS anymore. You have to copy this right here. Okay, so first we run and we load some important libraries or packages that will allow us to analyze data and visualize the data. So here you have to wait until that finishes. But as it works, we can go in here and change the name from what we have in the link to the name of the actual file and see what is it that we are doing next. We are dropping the ID column, and we transpose our data. And then we can just print the data. So here we just have to wait. Oh, okay, this is not working because I'm in Safari. Let me just quickly open this in Chrome, and now there it will work. Okay, so once we do it here in Chrome, all right, so let's do next. Okay. So here, by the way, we will be able to see once the packages are loaded, which packages are loaded already. So I'm just going to drag and drop this right here, drag and drop. Let's do run. So while this is executing, I'm just going to change this right here. Okay, and okay, now you can see that the packages are loaded, and I can click on run. So here it displayed all of the names of my rows and the value. I can convert those integers to floats, okay, and create the box plot. So if you click on run, okay, log is not defined, so we have not defined the log. Okay, so here we need to change the log to data final. And here we have our box plot. So a very similar procedure, but here you just have to drop the file here and then run the code in the browser. So for example, we did a box plot. Here we can do a histogram. So again, here we don't have, let's change it to data final. Okay, here we have a histogram. I guess here, instead of doing the histogram for everything, we need to select which one we want to do it for. And some other exercises that we can do. For example, here we are learning how to filter the values and do a heat map. We can also convert the data, and then we can do a color bar. So we can do a bar plot down here. So the objective for doing these types of exercises is to learn how to read these pieces of code that are very similar on one hand, because they actually do very similar things. They take a particular portion of the data and run a function, but they work a little bit differently between R and Python. So knowing how to navigate your code and changing the right parts of the code is the first part that we want to accomplish in these exercises. Okay, so this is what we will cover in today's session. We have another session coming up, so we're really going to have to switch over. But I hope that this was somewhat informative for all of you. We will continue to work through these exercises, and you can also try to work through the exercises on your own. If you find some syntax issues, then you can reach out to us, and we'll look into them. And in our next session, we'll practice a little bit more, and we'll try to prepare several examples so that we can more significantly show the differences between the input and the output data based on different settings that we employ in the code. Thank you for joining today, and we'll see you again next week.